Go ahead and get started. Thank you all for joining. Really excited to have Dr. Megan Kraft in to talk. I've had the pleasure of working with cougars a lot out in Yellowstone and some disease work, so I'm really excited about this talk, and I hope you guys are too. Before we get started, just a couple little housekeeping items. If you have any questions, feel free to put them in the chat, and then we'll have a Q&A after her talk that I'll help moderate. And if you want to hold a question and be brave and speak up at the end, we'd also encourage you to do that as well. So without further ado, it's my pleasure to introduce Dr. Megan Kraft. She's an infectious disease ecologist and associate professor here at the university in the EEB department. After completing her undergraduate degree in biology from Brown University, she took some time off in Africa before applying to medical or vet school. She fell in love with East Africa's wildlife, people, and landscapes, and ended up spending 6 years in East Africa, where she started a safari company and studied lions. In her late 20s, she began her PhD at the University of Minnesota and focused on disease dynamics in African lions. She completed postdocs and fellowships at the University of Texas, the University of Glasgow, and University of Minnesota's Institute on the Environment before starting her UMN faculty position here in 2011. She currently leads a lab aiming to understand infectious disease dynamics in animal and human populations. So with that, take it away, Dr. Kraft.
Thank you all, and thanks to the organizers for the honor and the pleasure to speak here today. So today I want to talk about understanding pathogen transmission in a solitary secretive carnivore, Puma concolor. So some call this the era of the Anthropocene. Our impacts on the environment are greater than ever. Environmental impacts include climate change, urbanization, habitat fragmentation, and changes in land use. These anthropogenic changes have implications for wildlife, including implications for wildlife disease. Wildlife, vectors, humans, and domestic animals are encountering each other in new places and in new ways. Transmission dynamics are changing between populations and can lead to disease spillover. Transmission dynamics are also changing within animal populations as these populations become more fragmented and experience changing densities. So how will we manage disease in the face of anthropogenic changes? So to understand how to manage disease threats, it is important to understand the basic process of transmission. For example, what conditions promote transmission from an infected to a susceptible individual? With this type of information, we can more effectively implement management strategies to stop transmission. So today I want to focus on a case study to understand transmission within wild populations. I want to understand transmission within Puma populations in a changing world. So specifically, I want to ask, how might pathogen transmission be shaped in human-dominated landscapes? I want to give a bit of a narrative about how my team, my lab and I have approached this through multiple lenses, starting off with serology, tackling it through contact networks and simulation modeling, and then ending with a story about pathogen genomics. I must highlight that this is the collective work of an awesome interdisciplinary team. We are a team of academic PIs and managers from state and national wildlife agencies. We consist of veterinarians, disease ecologists, landscape geneticists, and conservation biologists too. I come at this from the point of view of an ecologist. I'm the disease ecologist on this team. And I would be remiss not to mention that most of the work I'm going to present today is from my PhD students and postdocs, Marie Gilbertson, Lauren White, and Nick Fountain-Jones. So let me start off with a little bit about the host. So today I'm going to focus on Puma, or Puma concolor to be specific. Other common names for this species include the mountain lion, panther, or cougar. So Puma are solitary, secretive carnivores, and juveniles disperse from their mother between 1 and 2 years of age, and then establish their own territories. Puma have very large overlapping home ranges and avoid each other except for mating opportunities. Males have larger territories than females and engage in more aggressive behavior. There has been documentation of carcass sharing in some areas, but it is pretty unclear how often Puma contact each other outside of mating. Puma used to range across the Americas in the areas covered by yellow and red in this map, but now are restricted to the red areas. We have been studying Puma in the 3 areas represented by the black boxes. Let me zoom in a bit to those areas. So we study Puma in 3 areas that vary widely in their management of Puma, and as well as the demographic characteristics of those populations. So for example, in California, Puma live in highly fragmented landscapes, and their population's relatively stable there. Whereas in Colorado, Puma can live in more open, natural landscapes, and this population is even hunted for sport. We can contrast that to the endangered Florida panther that is so isolated it had to be genetically rescued from Puma from Texas. So that's a population that's growing. That population is also extremely isolated. It's not fragmented, it's just a small closed population. So wildlife samples were collected by our collaborators in the three study regions using different methods. Some use houndsmen and some use foothold traps or cage traps. And whenever a puma is collected, blood samples are taken, and these blood samples were then given to my colleagues at Colorado State University who conducted serology. We also obtained samples from Puma hit from car strikes. In terms of infectious agents, we screened for all the pathogens here in the left column. But today I want to focus on only one of those. I want to talk about feline immunodeficiency virus. The reason I'm choosing to focus on this virus today is because it is host specific and is transmitted by direct transmission. We are interested in better understanding how diseases transmit within a host population. So this seemed like a good pathogen to look at. It should be noted that this pathogen is apathogenic. So that's a mouthful. So what I'm trying to say is that this infectious agent doesn't seem to cause disease in its host and cause any harm. It also causes a chronic infection, which means once you get infected with FIV, you are infectious throughout the whole life of the individual. And it is unclear what the conservation implications of this pathogen are. Okay, so let me dive a little bit more into the serology part of this story here. Okay, so let's look at what's going on with this feline immunodeficiency in Puma. So let's look here at this red arrow where it shows FIV in Puma in the yellow, in all the areas, which is about 50% prevalence in California, Colorado, and Florida. So another reason we chose to focus on this virus is it's quite common in these 3 areas. When we plotted the FIV prevalence by Puma age, you can see that there is a gradual increasing the older a Puma is, the more likely it is to have been infected with FIV. This is to be expected with a chronically infectious pathogen. The longer you've been on the landscape, the more likely you can be exposed. But we did some modeling and found out that indeed there was no infection-induced mortality caused by being infected with FIV. Infected cats don't live any shorter lives than non-infected cats. We then measured the force of infection. So the force of infection is just how likely susceptible is to become infected with this pathogen. And we found some interesting differences. For example, in the graph on the right, you can see that males in general have a higher force of infection than females do, graphed on the left. You can also see that we have some differences between the different study sites, with Colorado potentially having a higher force of infection than the other two sites. But of course, your eye is going to be drawn to the descending blue line with the males in Florida. Here we see something different. Here, Florida males are much more likely to be infected when they are young than when they are old. We think this represents the competition, extreme competition for territory in this in this very closed population. In fact, males in Florida often die before the age of 3 due to intraspecific aggression. Okay, so the next step in checking out what's going on with FIV in these Puma was to check out if cases were spatially autocorrelated. With spatial autocorrelation, you might expect that Puma in one area might have the same exposure status and have it be different than Puma that are further away, who might have the opposite exposure status in a different area, so be more clustered. But what we found is that FIV exposure in Puma was not spatially autocorrelated. And I think this has to do with the fact that it's a chronic infection and it's in a wide-ranging species, and that these home ranges are really overlapping. I'll show you some maps in a little bit, but so maybe something like spatial autocorrelation just can't really be picked up using serology with such wide-ranging species. Okay, so to wrap up what we learned about serology, we found that FIV is common in all our sites. Individuals are likely to get infected throughout their whole lifespan. FIV doesn't seem to cause mortality. And for a wide-ranging species like this, geographic epidemiology didn't really give us much information. So we decided to dive into some more approaches to try and figure out how pathogen transmission could be happening on these landscapes. So we attempted some contact networks and simulation modeling. So with my previous work on more social species, such as raccoons, African lions, and Hawaiian monk seals, you can use behavioral observations or proximity logger data or data collected from animal movements like GPS and VHF to construct what we call a contact network. So in these contact networks, the individual animals or groups of animals can be seen as the dots on these networks, and the lines between the dots can be edges representing contacts. And then what you can do is you assume that if diseases are transmitted by direct contact, that some proportion of those edges or contacts could lead to transmission events. And that's how you could end up with a transmission network that could represent how diseases spread through those populations. Okay, so let's say, well, let's try this for now this solitary secretive species. So first off, we tried this in California with using global positioning sensing color data. This was work done by Lauren White. She looked at 30 individuals over a 10-year period who had GPS data. These GPS colors were collecting fixes every 5 to 15 minutes, and the path of one individual can be shown on the right. So she said, okay, let's try and make a contact network based on spatial temporal overlap. So whenever we get an individual within the same time and space as another one, we'll call it a contact. Well, when she looked at the data, she found that the number of pairs found, the number of pairs on the y-axis, minimum distance on On the x-axis, the number of pairs found in close proximity were frankly just the mother and offspring. So from a disease transmission point of view, that's not actually that interesting for us. So she tried another approach. She also tried spatial overlap. So here you can assume that the more different Puma's home ranges overlap, the more likely they might be to have a close encounter and transmit a pathogen. And so you can draw edges or links between the different Puma if they have spatial overlap. So indeed, Lauren found some spatial overlap between these different Puma. But with this map, she actually has all the data aggregated. So we're not even sure that these Puma were on the landscape at the same time. So that's a drawback of trying to estimate contacts this way. When she made the two networks using the different approaches, we got quite different results. Ideally, for a directly transmitted pathogen like FIV, you want to use spatial temporal overlap because you need to be in the same place at the same time to have a direct contact. But when you get a network that looks like this, it's hard to think over how you could actually get like a 50% prevalence when the network's so disconnected. Okay. So we said, okay, maybe using empirical movement data isn't a good way to estimate contacts in Puma. Luckily, Lauren has enviable programming skills and could do a simulation study instead of using empirical data. So she built a model where these different colors represent territorial individuals who move around the landscape, leaving a scent mark that can repel other individuals, thereby forming territories. And she found out that territorial behavior can dampen disease prevalence with that spatial structuring, but it can also increase pathogen persistence. So that was a neat finding. It wasn't directly relevant to the California Puma system, but it can explain some disease dynamics we see. All right, well, maybe VHF telemetry data might work better. So let's look into some data from Florida. So it's important to note that the Florida panther lives in a highly seasonal environment with dry seasons and wet seasons. So Marie Gilbertson checked out 10 years of VHF telemetry data from Florida. This telemetry data was collected from aerial surveys 3 times a week for 10 years, where they know where all these cats are. So amazing wildlife data. Let's see if we can make some contact networks out of it. So here's an example of what she created. In the network on the left, it represents spatial overlap between Puma in the dry season. And on the right, it represents spatial overlap between Puma on the wet season. You can see that Puma in the dry season are arguably more connected. They have more spatial overlap. And we found this to be true across all the different years. So here the dry season is represented by the beige color and the wet by the greenish color. And we found that the dry season generally had more connectivity across the years. So what happens if you introduce a pathogen on these sorts of contact networks? How might it spread in the wet season and the dry season? So Marie was pathogen agnostic here and simulated a bunch of different types of pathogens with different infectious periods, et cetera. And here's some of the results she found. She found that if you introduce a pathogen in the dry season, when the network's more connected, it makes sense that there can be more individuals getting infected. In fact, we found that up to like 2 more individuals could get infected if the disease was introduced during the dry season versus the wet season. And I think the main take-home message from this type of study is we still need to remember that there can be seasonal drivers that can change behavior and contact patterns when we think about disease transmission on landscapes. All right, so that's kind of what we found out for the contact networks and simulation modeling. It turns out that figuring out, it turns out that it's really challenging to utilize this commonly collected GPS and VHF data for making contact networks, because these interactions between the Puma are so rare since they're solitary and secretive. And we really just didn't have the fine scale resolution needed to make contact networks based on this type of data. But what can pathogen genomics tell us? What information can we glean by looking at the pathogen itself? I have a few stories I'm going to try and tell here under this section. So the first is, so you might have noticed that the pathogen I focused on so far doesn't cause disease in the host. This is intentional here, and Marie Gilbertson wanted to go out on a limb and try and figure out if using apathogenic infections could predict disease dynamics. Can we use the genomics of a virus to determine transmission pathways? And those same transmission pathways might be utilized for transmission of a pathogen that might be more virulent, or might be one we might care more about for conservation. So more specifically, she wanted to know, can apathogenic infections predict pathogen dynamics? For example, if you had a transmission network based on phylogenetic relatedness of, say, a virus, could you figure out what environmental and ecological factors predicted that transmission, and then apply these rules of transmission to create a virtual contact network? Then you could simulate transmission of a related pathogen on that virtual contact network and look at different disease, do this stochastically and look at different disease outcomes. So we thought we would try this in the Florida Panthers. Potentially, they are a model system to use this in. These Florida Panthers have been well studied. We have movement data on them. We have loads of landscape covariates and landscape layers. We have a complete pedigree of who's related to who there, often too closely related to each other, but we know how related they are. And we have serology samples and disease data. For this little story, I'm just going to talk about feline immunodeficiency virus. Remember, that's the apathogenic one. And a new virus, I want to talk about feline leukemia virus. So as I mentioned, FIV is apathogenic, directly transmitted, causes a persistent infection, is species specific, and has a rapid mutation rate. Feline leukemia virus is pathogenic. It's directly transmitted, so same mode of transmission as FIV. And there was a well-documented outbreak in panthers between 2002 and 2004 that killed, I think, about 5 panthers and infected many more. And we can't study the transmission of feline leukemia virus very well because panthers that get it, they die, right? So here we're trying to figure out if FIV can be like a proxy for how feline leukemia virus might transmit on this landscape. So with FELV, there's still ongoing transmission and spillover happening in Florida right now. And these panthers are actively managed for feline leukemia virus, but there's many uncertainties about how they are being managed. The managers are uncertain about how they're managing them. So we wanted to test if apathogenic viral infections could act as proxies of transmission relevant contact. And here's what Marie did. It's always challenging sharing half of someone's PhD work in one slide. So let's see what I can do here. First, she wanted to know who transmitted FIV to whom. So she had these genomic sequences of this fast mutating virus and could make a transmission network. So that's this network on the right. So this is who actually transmitted virus to whom. Then she used exponential random graph modeling to ask what factors structure FIV transmission. And she could simulate other networks that had those similar properties and make them be relevant to the population of Florida panthers at the beginning of the 2002-2004 outbreak. So kind of parameterize that with the population size and the gender ratio, sex ratio at the beginning of the epidemic. Then she could simulate this more virulent pathogen, FELV, on these networks and ask how do these FIV rules of transmission predict FELV transmission dynamics. And she also asked, how do these FIV-based predictions compare to 3 other types of models? So this FIV-based model is one based on this genomics, but potentially you could just spread disease through this population, for example, assuming that these cats were all well mixed, or there's other simpler models to use. This is what the observed feline leukemia outbreak looked like in the mid-2000s. The blue dots are the negative panthers, the red dots are the infected panthers, and in the north, you can see a spatial cluster of cases there. So FELV in this outbreak did show some spatial autocorrelation. So what she's going to do is take the FIV network, make it to represent the panther population at the time of this outbreak, and then simulate feline leukemia virus on that many times and see if she can replicate this pattern of this outbreak pattern. So when she figured out, so with what factors structure FIV transmission, this is what she found. Here is the actual transmission network of FIV in the Florida panther population. The red nodes are the females, the blue nodes are the males, the circles are the adults, the squares are the juveniles, the weight of the edges represents our confidence in that being the transmission route. And it basically came out to that FIV in Florida panthers is mostly transmitted between individuals that are close together in space and that are from the same age class. And when she simulated outbreaks using this model based on FIV with similar age class and you're near each other in space, she could do a good job of simulating the spatial clustering found in the outbreak. So for these graphs, here's our FIV-based model on the x-axis. Here's our cluster radius on the y-axis. And her FIV-based model was closer to the observed outbreak, which is the red line, than another model, which was the overlap-based model. And similarly, the observed or expected cases were closer in the FIV-based model than the overlap-based model because it was closer to this red line representing the real-world data. So when she was evaluating model performance between these different types of models, she thought the FIV-based model worked the best because it could represent spatial clustering better than these other types of models could. So in summary, for that section, we thought that drivers of transmission for an apathogenic virus can predict transmission dynamics for a pathogenic virus. So the obvious next step to take there is to say, well, hey, what's the optimal management strategies for FELV for feline leukemia virus and Florida panthers? So panther managers have a couple of options of what to do to manage an infectious disease. They can vaccinate, but there's questions about boosters. There's questions about efficacy of vaccines. I wonder if some of this is resonating with some of the questions we're having about current coronavirus pandemic, right? There's even questions about how to model vaccinations. So do vaccinations induce binary immunity where some individuals are protected and some aren't? Or does vaccination induce partial immunity where individuals are kind of immune, but not all the way? So with this study, Marie went with the vaccination inducing partial immunity, which is kind of a new thing to do with wildlife studies. Other tools managers have include test and removal. So this literally means you give a test to an animal. If they're an endangered species like this, you can take them off site, put them in a pen, let them recover, and then introduce them back to the population. Okay, so her methods for this was she simulated a contact network. She simulated FELV transmission on this network with no interventions. And then this like complicated, I guess, factorial design about testing different interventions, where she would test proactive vaccination. So that means vaccinating before an outbreak, reactive vaccination once an outbreak has been detected, and then reactive test and removal, and a bunch of different scenarios as well of different types of boosting or proportion vaccinated. And then she would compare these outcomes from the different types of treatments to the one with no interventions. And we're trying to figure out which combination of interventions work best. We came upon an interesting, I guess, paradox, and found that, I guess, ironically, proactive vaccination with partial immunity could cause more individuals to be infected than doing nothing at all. Let me walk you through that a little bit slower. So these graphs here, the x-axis of these graphs show the number of mortalities, and these are a histogram of simulation results showing the distribution of number of mortalities. And on this axis, we're showing the change in proportion vaccinated from very low to very high vaccination. And across the top here, we have the proportion receiving a booster going from low to high as well. And there's this interesting, the red line shows the median mortalities with no interventions. So that's the do nothing scenario, like right here. And then the blue line shows the median mortalities with a given level of proactive vaccination. So we found this interesting parameter space with low vaccination rate, where there were actually, in some cases, more mortalities due to low vaccination than doing nothing. And we think with this partial immunity that we assumed in our model, it allowed these individuals to be on the landscape longer, be exposed, and get infected later on in the outbreak, thereby giving a chance to infect somebody else later in an outbreak. So kind of dragging the outbreak on longer, allowing more mortalities to happen. Anyway, this is an interesting counterintuitive result that we found. So that's the paradox, that low proactive vaccination can worsen some epidemic outcomes. But the good news is she also found some synergies as well, that proactive vaccination and reactive test and removal or vaccination can reduce mortalities in this population. So the big take-home message here is that we think that improved proactive pathogen management is possible and can reveal unexpected outcomes from management interventions. All right, I'm going to take a drink of water, move on to a story from Colorado. Okay, so switching gears a little bit, but still staying under the focus of looking at pathogen genomics. We're going to dive into a case study in Colorado. And this is work led by my postdoc, Nick Fountain Jones, where he's asking, how is FIV moving on the landscape? So when you zoom into Colorado, we had data from 2 very different regions in Colorado. The one on the west, we're calling the unbounded region. This is an area kind of natural, and the Puma, I'll get to this later, are actually hunted for sport here. And on the east here, we have this, what we're calling the wildland urban interface. This population of Puma abuts to the Denver metropolitan area in the Front Range. So you got Denver, Boulder, Fort Collins here. Okay, and so what I'm going to show now is we're going to compare the genomics of the host to the genomics of the virus. And we're going to see if viruses flow on the landscape similarly to host gene flow on the landscape. Okay, so here's what happened when we look at the host genomics. So in the unbounded area to the west, we found that host gene flow was facilitated by forest cover. So hosts are moving in areas with forests. And we found pretty much the opposite in this wildland urban interface. We found that host gene flow was impeded by impervious surfaces. So hosts are not moving around urban areas and not breeding in urban areas. So you can take those results and make a Puma relatedness cladogram between our 2 different regions. Here, the purple is the wildland urban interface, and the green branches represent the unbounded region. And you can see that, as you'd expect, Puma in one region are more closely related to those individuals than they are to the other region. And there's a couple of immigrants in here, which are kind of cool to pick up too. But let's see what the virus looks like. Are we going to see the same relatedness for the virus? So here's when we plotted how the strains of FIV are related to each other. And similarly, we do see some regional clustering. So the wildland urban interface virus strains cluster together, and so do the unbounded region virus strains. Okay, well, let's see if this stuff matches up. Well, it does not. These don't match perfectly. This is called a tangogram, and you can see why it's called a tangogram. So these don't, the viruses and the host relatedness don't match precisely. So something else must be going on about how these viruses are transmitting on this landscape. I'm going to put this into a video. Well, Nick put this into a video. Let me show it to you. So this is the unbounded population. This is the area to the west. And you're going to see some blue dots popping up to represent uninfected cats, red dots representing infected cats. And we're going to have some lines showing how the virus is moving up on the landscape. I'll show this twice. I wouldn't worry about reading the words. Okay, let me play this again. I mostly want you to look at how you mostly see transmission moving with kind of short jumps and occasional few long jumps. Okay, and then let's compare that to what is happening at the wildland urban interface. So we're going to see what's happening here. Okay, here's Denver, Boulder, Fort Collins. And the main takeaway message here is I want you to notice how transmission really is just going in small jumps north and south up in this region, north and south. These cats and the viruses are kind of trapped between the mountaintops on the west and the urbanization to the east. Let me play this one more time. Okay, so we think that urbanization is impacting FIV spread. In this unbounded area to the west, we find that those viruses are actually best transmitted along roads. So we find that the FIV strains are more related when they are closely connected by dirt roads. So these these Puma seem to be traveling along dirt roads and spreading disease that way. In the wildland urban interface, we see a different story where FIV strains are more similar. The closer the cats are located together, the more related these individuals are, and the less amount of impervious surface there are. So that's why we think urbanization is actually impacting FIV strains. We think that the cats in this area basically have nowhere to disperse to, and so they're having to settle closer to their close relatives, and that's why transmission is happening that way. And yeah. All right, so we're going to look at these two regions now through a different lens. Before I was looking at urbanization, not urban areas, and now I want to look at Colorado through a different lens of hunting. So Puma are hunted in Colorado, and they were hunted in this treatment region before 2004, and it should be noted that hunters prefer males. And then between 2004 and 2009, managers did an experiment and they removed hunting from this population. And then after 2009, they resumed hunting. So we're calling this the treatment region because management was modified from hunting, no hunting, to hunting. And this area to the east, we're calling the stable region. Here, there is very low hunting of Puma throughout this whole time period, and it was like consistently very low. So we're gonna call that the stable region. This paper just came out last week in Nature, Ecology, and Evolution, so we're very excited about it. Okay, here's what we found when we compared transmission networks between these two areas. We saw some differences in viral transmission, whether areas were hunted, whether there's a change in management from hunting in this treatment region or in the stable region. So in the treatment region, you can see that transmission is dominated by these males. They're in blue. As compared to the stable region, where it's more between males and females, if not maybe even dominated by females. And in this treatment region, our estimated R0 is 4.1. That's double the R0 of in the stable region. And that means an infectious individual is more than 2 times is likely to infect 2 more individuals than they are in the stable region if they're in this treatment region. When we plotted this out through time, in the treatment region, you can see that most transmission events take place during this no hunting period. And it kind of slows down during when hunting was resumed in the hunting period. And you can contrast that to what happened during the stable region, where transmission's kind of burbling along slowly through time. So we think that hunting, in fact, caused differences in viral transmission. And more specifically, the release from hunting is what caused these changes. So we saw that the release from hunting increased the viral population growth rate. So here on the x-axis, we have the year growth rate. On the y-axis, look at the black line. This is the FIV growth rate through time. So hunting, stop hunting, and then the viral growth rate increases. And then once hunting resumes again, it decreases. And this trend was statistically correlated with the male population size here. So what we think happened with this story is that males were being hunted in this treatment region. They stopped hunting, and therefore males were allowed to persist in this region. Maybe they came in via immigration, or survivalship just went up. And then with that increase in male density, more transmission events could take place. And then once the hunting started again, males population size reduced and transmission decreased. So in summary, from the pathogen genomics, here we found that at the wildland-urban interface, transmission was more spatial and between close relatives than in the unbounded region. And in terms of disease management, this might mean that if you have an outbreak near an urban area, if you want to try and manage it with vaccination, you might be able to do targeted spatial vaccination. For example, if like feline leukemia came in there, you could treat in a smaller spatial region. In terms of hunting, we had wondered how this virus, if this virus flowed the way that host genes flow. And we found out that viruses can give us more resolution than host genes and can even reveal sex-specific differences in transmission. And so after a demographic perturbation like hunting or cessation of hunting, if targeted vaccination were to be used, maybe a certain sex could be targeted. So in conclusion, we are going to continue to face pressing disease issues like these. In this era of the Anthropocene, we need to continue to be creative in how to manage wildlife disease by developing new analytical approaches, leveraging these decreasing costs of high resolution molecular data, capitalizing on new animal tracking technologies, and partnering with a wide range of stakeholders with complementary and varied expertise. I personally feel that folks in conservation sciences are especially well equipped to tackle these pressing and emerging issues. With that, I'd like to thank my funding sources, my wonderful collaborators for making this research fun and personally rewarding. And I also thank you for your attention. I hope I have some time for some questions, and thank you all.
Great, thanks, Dr. Kraft. Yeah, that was a really great talk. And I think there's a lot of really broad applicability to think about too, kind of as you said, for even for people in disease transmission. So it looks like we have one question in the chat, but maybe and I have a couple questions too, but maybe I'll open it up to any other brave souls that might want to unmute and ask a question directly first. Give it maybe 10 to 15 seconds. All right, I'll go to the chat. So you might have answered this after the question, but Barbara asks, how is the virus transmitted? By saliva, urine, et cetera? And then kind of adding on to that, is the disease only transmitted directly from cougar to cougar, or is it possible to persist in the environment for a period of time, or move through other felids like bobcats that might also be on the landscape?
Great point. So FIV, we believe, is transmitted by direct contact. We're pretty sure fighting, mating, biting are possible routes. We're less clear if like saliva transmission from mother to offspring is another mechanism of it, but it seems like close contact should work. We don't think there's environmental persistence from FI with FIV. FIV, Puma can get FIV strains from bobcats when they eat the bobcat. So you can find bobcat FIV in a Puma, but they don't seem to transmit that onwards. And we excluded those bobcat FIVs from our analysis. Feline leukemia virus, on the other hand, spills over from domestic cats. And so at the Florida Panther story, we think that those Puma are eating domestic cats they find on golf courses around there. And that's how that Panther population is getting feline leukemia virus. It's a spillover, but then there indeed is onward transmission of feline leukemia virus from Panther to Panther once they get it. In terms of environmentally transmitted pathogens, we were making assumptions with some of those like spatial overlap models that there are other pathogens that you can pick up through that have a better environmental persistence that you could get using indirect routes. Kalichi virus, or you guys were talking about, toxo some more. But yeah, there's other pathogens that can be transmitted indirectly. I didn't focus on those today.
Yeah, great, thanks. And then I had one question. So when you were talking about the kind of treatment versus stable populations, the one urban, one kind of more in this wild setting.
Yeah.
You mentioned that the treatment group had about double the infectious rating, double the R naught than the stable one. Do you think that's due to like higher densities in the treatment versus the stable? Because I know you mentioned that the stable, they were kind of pushed to live closer together. Is that more just in that kind of north-south boundary, but they just still have a much lower density and fewer contacts than say in the treatment population?
Great questions. I did not give you guys the full story there. It's hard to in that time period. So we actually think, ironically, we think the densities of those cats are similar in those two regions. One is, I guess, is stretched kind of like this, but the other one's like more square and can have lots of immigration and emigration out of that area. So we think that in terms of populations, those two areas are pretty comparable. When I brought the density thing in, it was more to like, once they stopped hunting, the number of males could increase, and transmission also increased with the number of males. So if we want to blame like males for aggressive interactions, that would be like a density dependent transmission thing. I have a hypothesis that when males got to survive on this landscape, they didn't have any like older males telling them how to behave or where to go. And so it was like these young males going rogue, fighting and trying to figure out like a new territorial order. But the managers on the ground say they can't support my hypothesis that that's what was going on. So we're just very vague that, as the male densities increase, transmission also decreased, but we can't exactly figure out why. The managers say that they don't know of Puma that can get into fights and survive those fights. So they are befuddled as to how this virus is transmitting themselves as well.
Yeah. Interesting. I'd be interested to look at, like, say, for my kind of study system out in Yellowstone, where you have a lot of cougars that are living, say, in the national park, where we have most likely a much higher density in proportion of the population that's males compared to just outside the park where there's a lot of male hunting. It'd be interesting to kind of compare the disease transmissions across that kind of national park boundary. But yeah, thanks.
That'd be pretty cool. Like, part of me wants to blame it a lot on the males, but indeed males were behaving properly over in the stable area too. So
yeah,
it almost seems like it was the change in densities or the change in social structure that set some sort of perturbation in a weird way. I don't know. Yeah.
So it looks like we don't have any more questions in the chat. Is there anyone that has any questions? Feel free to unmute or drop them in the chat if you have them.
I'll ask a quick question to keep this going.
Good.
Megan, when you were doing the simulations of the spread, this is I guess in the third part of the talk. I'm not sure which part it was, but you had the simulations of the cougars. What are you using? Do you do parameter sensitivity for contact rates and probability of transmission? Because I just wonder, I always wonder when we're trying to estimate these things from, empirical data and then simulate them, lots of animals must come and like pass by each other and not ever transmit anything or
Right.
actually have an event. So how do you actually deal with that in a sensitivity analysis of your simulations? And did you see any effect on the rate of spread? How sensitive was it?
I hope I'm going to answer your question correctly. We, I think for that study, we assumed we knew what rules to use to create edges between individuals. And then we did a lot of stochastic types. We made that population stochastically many times to just make sure it wasn't like that one realization that happened to have it that way. And so we simulated a population that matched how FIV would have transmitted. And
Right,
so you add some stochasticity in there.
So because you had those edges that were like the ones you were really confident on and less confident on. Yeah. I was just wondering how did the level of confidence, I guess, affect how things were perceived or analyzed?
That's a good point. With those, we ended up not using those confidence bins and having to go with, if you were ever connected, we're going to connect you with that. But part two of your question at some point, I think, was, and then basically you're just flipping a coin with, if there's an edge there, does a transmission happen between an infected and an susceptible during that time step? And you can like weight that coin as you need to make it more transmissible or less transmissible given a contact.
Okay.
Is that helpful?
Yeah. I guess what I'm always confused about with disease models is that, if there's some probability of infection, it will get there eventually, right? It will infect everybody eventually, like through time. It's just a matter of, is it a reasonable amount of time if there is some level of connectivity, right? And so obviously
if it's a SI, if you're infectious forever, yes.
Yeah.
But if you're
removed,
if you're coronavirus and it's only a week or something, then it's just whether you happen to be connected during that moment you were infectious or not.
Right.
So that's why some of these like chronic infection, like the FIV, we only just ran it that one season because it's kind of boring to run it for 10 years because everybody would get it over time.
That answers my question.
Yeah.
Okay, thank you.
Yeah. But James, I didn't explicitly point this out, but there's a big gap between what movement ecology can tell us and what disease ecology can do. And like just getting that data to integrate is really a frontier we got to work on. Like it's that movement data is great. And then we've got this disease stuff, but the parameters you get from movement models don't really feed into the disease models well.
Right.
So,
well, I think that's what you showed in the first part of your talk. And it's definitely something I think you and I and others have been challenged with, is that how do we take that information of how these animals move and translate it meaningfully into the disease ecology? And I don't know, like you kind of show that it's tough. It's harder than we think. And it's hard to know how to translate that into population level parameters.
That involve direct contact, right?
Yeah, exactly.
Yeah.
Awesome. Well, I think we're right past 1. If we don't have any other questions, I think we'll end it here. Yeah, thanks, Dr. Kraft, again for a really, really interesting talk. Thanks for joining us.
Thank you all for your attention and for coming and spending Friday with us.
Yeah,
Okay,
have a good
take care.
weekend, everybody.
Follow up on email if anybody wants to.